Okay, so today we have to talk about how do you get from genes made of DNA to protein made of amino acids. So I'm just going to step out of the way so you can have the full screen. And let's remember what DNA is. DNA is a code. It consists of those, remember those bases A, C, T, and G, but it's located in the nucleus of the cell. And if you remember, proteins are made on the ribosome, which happens to be in the cytoplasm. What are you going to do? How are you going to get from the nucleus to the cytoplasm? Well, you have to go through a process called transcription. And transcription is when you make a messenger RNA copy of DNA. You may recall we talked about messenger RNA, you learned about that, or mRNA. That is the one that carries the message or the copy of that DNA. And this happens at what is called the promoter region, and there's an enzyme called RNA polymerase, which makes RNA, and it's going to help add all those bases. The messenger RNA itself is going to base pair with your DNA, so wherever there's a C, there's going to be a G in your messenger RNA. You can see in the picture. There's one difference, however, just to make it interesting. Instead of thymine or T, in messenger RNA you have uracil or U instead. So that's one difference. Another difference is it is a single strand instead of a double strand, because you only need one copy that's going to leave. Why make a copy? Why not just send that DNA out? Well, if you think about it, if the DNA is your only copy and it's really important for staying alive, if you send it out into the big bad world of the cytoplasm, you're not going, it may not come back. It might get destroyed. Then what? Now you're sunk. Kind of like when you're in the library and they have those reference books and they won't let you take them out because they're really important. So what do? You make a copy at the copy machine and you take that. This is kind of the same idea, that you take out your messenger RNA as a copy and leave the original so that it's there for use again in the future. Once you get out of the nucleus, the messenger RNA goes out, it's going to help form the ribosome with the other two RNAs that you learned about, ribosomal RNA or rRNA and transfer RNA or tRNA. These come together, and this is a nice little graphic of the ribosome. You can see all the bases on the bottom there. It starts with UCC, and that's running through the ribosome, which has two parts. There's that small base, and then there's the upper portion, which is larger, and it has those T-shaped transfer RNAs in there. And you can see the amino acids, those are the letters M and S, those are at the top of the transfer RNAs. So every 3 bases of messenger RNA codes for one amino acid. It is called a codon. So you can easily remember that. tRNA has an anticodon, and this anticodon will base pair with the codon. So you can see there's a lot of base pairing going on. And this is how you know which amino acid to get. If you didn't have this little system, you might have a mix-up on what amino acid goes where. Each tRNA is also highly specific for one specific amino acid. And there is a start codon, which tells you where to begin translating and making your protein. There's also a stop codon that will tell you that this is the end of the polypeptide, or the many units, which is basically a protein. This diagram shows you have your messenger RNA, that's that blue strip. Then it shows the ribosome is green. You have the tRNAs aren't shaped like a T in this case, but you can see all those amino acids. It says MET, PRO, ASP, GLU. You don't have to know all the amino acids by heart. Those are the 3-letter codes. But you can see that they're forming a long chain. You're going to try a little translation yourself and practice with transcription and translation.